When you boil it all down, every single living thing on this planet has the exact same fundamental challenge. How do you copy your genetic blueprint accurately and pass it on? It's the most critical job there is, and as we're about to see, the strategies for getting it done are incredibly diverse, each one sculpted by its own unique evolutionary pressures.
So yeah, how does life make more life? Whether we're talking about one of our own cells, a single bacterium, or a virus, the end goal is always the same: replication. But the way they get there, well, that couldn't be more different. We're going to dig into the what, and maybe more importantly, the why behind these very different paths. Okay, let's get into it. Just take a look at the contrast right here. On the left, you've got this intricate, almost beautiful dance of mitosis in eukaryotic cells. It's a strategy that's all about control, about precision, which is absolutely essential for complex organisms like us, where just one mistake can lead to disaster, like cancer. And then, on the right, you have this teeming, chaotic-looking colony of E. coli. Their strategy isn't about some elegant choreography. It's about pure, relentless speed. It's life in the fast lane, where you're constantly fighting for resources.
So, to really get our heads around these different approaches, here's the game plan. We'll start with the eukaryotic blueprint, our baseline for precision. Then we'll see how viruses completely hijack that system. After that, we'll shift gears to the totally different world of prokaryotes, get into the nitty-gritty of how they're so fast, and finally, we'll pull all three strategies together.
Right, let's kick things off with the system that's the foundation for all multicellular life, the superregulated checkpoint-heavy cycle of an animal cell. Just think of this as our gold standard for doing things carefully and getting it right every time.
So, you know the classic phases: G1, where the cell grows, S phase for DNA synthesis, G2 for the final prep work, and of course, M for mitosis. Now, the whole show is run by these little molecular engines called cyclin-dependent kinases, or CDKs. They can't do anything on their own, though. They need to team up with their partners, the cyclins. These CDK-cyclin complexes are what flip the switches, pushing the cell from one phase to the next. But, and this is key, only when all the safety checks, the checkpoints, give the green light.
And a major player here is CDK1. When it finally pairs up with its partner, cyclin B, they form this complex that used to be called maturation promoting factor, or MPF. This is the complex that gives the cell that final, decisive shove over the cliff, from G2 right into mitosis. The big takeaway here is that the whole system is built on this really strict sequence of turning things on and turning things off. It's a beautiful adaptation for making sure the genome stays intact.
Okay, so we've got this perfectly balanced system. Now what happens when an intruder shows up? This brings us to our second strategy: viruses. They don't bother building their own factory, they just take over the one that's already there.
Viruses are really the ultimate parasites, aren't they? As obligate intracellular pathogens, they have zero machinery of their own for making copies. They have to take over the hosts. And a massive part of their playbook is to deliberately mess up that elegant cell cycle we just talked about, to break all the rules and create an environment that's perfect for just one thing: making more viruses. Now this is where it gets really clever, because there are two totally different tactics for taking control. Some viruses, like coronaviruses, actually want to slam the brakes on the cell cycle. They can ramp up production of proteins like P21, which is a powerful cell cycle inhibitor. Others, like influenza A, go after the retinoblastoma protein, PRB, to stop the cell from moving from G1 into S phase. So why would they do that? Simple. It's to stop the host from using up all the nucleotides and polymerases, freeing up the entire pool for the virus to use itself. It's basically a hostile resource grab.
But then you've got other viruses that do the complete opposite. Small DNA tumor viruses, like HPV and adenovirus, need the host's DNA replication machinery firing on all cylinders. So their viral oncoproteins, like E6 and E7 and HPV, are designed to seek and destroy the cell's main tumor suppressors, P53 and PRB. This essentially cuts the brakes on the cell cycle, forcing it into S phase, where the host's machinery has no idea it's now just a factory for copying thousands and thousands of viral genomes.
Okay, so we've seen precision control and we've seen a hostile takeover. Let's completely change gears and talk about our third strategy, which is a whole different ballgame. We're now entering the world of the prokaryotes.
I mean, this table pretty much says it all, doesn't it? A human cell takes about 24 hours to divide. E. coli, 20 minutes when conditions are good. That is a mind-boggling difference in speed. Bacteria are tiny, they don't have the cyclins and CDKs that we rely on, and this is huge. Their cell cycle isn't this neat sequence of separate events with checkpoints. Nope, it's a continuous,
overlapping process that is built for one thing and one thing only: exponential growth.
So how on earth do they pull off this incredible speed? Well, it's not just one thing. It's how they integrate everything: replication, segregation, and division. So it's all happening at the same time. Let's look at the mechanics. Unlike us eukaryotes with all our different origins of replication, a bacterium like E. coli keeps it simple. It's got one circular chromosome with a single starting point called oric. Replication kicks off there and then proceeds in both directions at the same time, kind of like two zippers moving away from each other around a circle until they meet at the end. It's just beautifully efficient.
And this right here is the absolute secret sauce to their speed. When there are plenty of nutrients around, E. coli doesn't even wait for one round of replication to finish before it starts the next one. It just kicks off new rounds from the new ori sites that have just been copied. This is called multi-fork replication. It's an amazing trick that means by the time a cell is actually ready to divide, its daughter cells will inherit chromosomes that are already halfway through the next round of replication.
So once the chromosomes are copied and pulled apart, the cell has to split in two. And the star of this show is a protein called FTZ. What's so cool is that FTZ is actually a homologue of our tubulin. It builds this structure called the Z-ring right in the middle of the cell, which you can see glowing green in this image. This ring is the master scaffold that recruits a whole team of other proteins, the divisome, to come in and build the new wall that pinches the cell in two.
So, to wrap up the bacterial cycle, we think of it in three connected periods. The B period is from birth to the start of replication. The C period is the time it takes to actually copy the chromosome. And the D period is the time from when replication ends to when the cell divides. But the real takeaway is that when things are growing fast, the next B and C periods start way before the current C and D periods are even done. The processes are totally layered on top of each other, creating that incredible symphony of speed.
So we've now been on a tour of 3 very different worlds, each with its own clever solution to the same universal problem of making more of itself. Let's just pull it all together.
So what have we seen? First, the eukaryotic strategy. It's careful, it's sequential, it's loaded with checkpoints. It has to be, because it prioritizes accuracy above all else. Second, the viral strategy. It's a hostile takeover, pure and simple. It breaks the host's rules for its own selfish gain. And third, the prokaryotic strategy, a masterpiece of raw efficiency where everything is happening at once, all geared for maximum speed to outcompete everyone else.
But look, these differences aren't just fascinating details for an exam. They have huge real-world consequences. And that leads to a final, really important question. Given that proteins like FZZ and the rest of the bacterial division machinery are essential for them but don't exist in our cells, how can we use that against them? This very difference is one of the biggest frontiers in the hunt for new antibiotics. Drugs that could kill bacteria by specifically shutting down their unique way of dividing while leaving our own cells completely alone. It's definitely something to think about.